believe that for now, there's no question about you are following the step one after the other. And once you are able to follow the step, I want to promise you that, honestly speaking, you could also be somebody that will be teaching others. Why is so? Let's go back to where we were. That is still under Galaxy, Abi.
Yes, sir.
Okay. So Galaxy. So you now look at one thing you should know if you want to run Procar, and you can see the full meaning of Procar. That is from prokaryotic gene annotation. That is what you want to do. So that means if you cannot make use of it for any eukaryotic, you cannot use prokaryotic. For example, if you are working with yeast or you are working with human sample, that is you cannot use procar because it is not for prokaryotic. So you need to actually master these tools and know what specific tools they are used for. It's just like somebody now normally from your tools you are disturbing now. Somebody, when we are talking about tools actually means that if not because if you want to sew cloths, what are those tools that you are going to need? Needle and what? Thread. So if you cannot see needle, can you make use of nail? So that is the idea behind all these bioinformatics tools. There are specific tools that are meant for specific purpose, and you need to know it. And that's the reason why that when you, just like I told you, when you encounter problem, maybe one tool is not going, it could be because every day and every time they normally upgrade their system. So let's go to Procar, just like I said. So you will see the last results on space on data sets. Make sure it is the last one you run on your system. On my own system, it is 238 and 237. Okay, and I also um make enquiry about the difference between scaffold and contigs from where? From what do we call it? ChatGPT. So which actually explain the meaning of the two and differentiated between the two for us. Okay, so where you are going to focus is that locus tag prefix, which is optional. So you can just tag your own, tag it anything. For me, I will tag it as a annotation. Okay. Then locus tag increment, leave it as it is. GFF fashion, leave it as it is. Leave the defaults. Every other one, leave it at default. Sequencing ID, I think it is 1773. That is for if you are dealing with, depending on the species of organism you are dealing with. But for Mycobacterium tuberculosis, it is 1773. So then the genus name is a Mycobacterium. Abi, what happened? Something is really wrong. I have to deselect all this. So it's like my cursor. Okay. So you leave that under select value. Leave that as it is. So it is going to run as a default. So which other one? So after Mycobacterium tuberculosis, the strain name, which is also optional, we can leave it as it is. Plasmid, leave it as it is. Under kingdom, it is a bacteria, leave it as a default. Now go on to run the tool. Run the tool. So and watch. So you will see that it has a change. The other on the right hand, you will see Proca on database. It's running already. So you wait until it has actually finished running. Now let's go to the BVBRC interface. Let's see how it is going to look like. I said you should register. So let's go to BV/BB /BRC. Okay. So open it. So and you can see, you click on, since we are working with bacteria, click on bacteria. So you are going to type Mycobacterium. Mycobacterium tuberculosis. Mycobacterium tuberculosis. Then you let it search. Please, you have to be monitoring your Galaxy. Check your Galaxy if it is still running. Okay. Now, on this our spade again, still under Galaxy, I want you to look at what do we have on Galaxy. Click on this view. Let's see the way it is going to look like. Okay. You can see all these are already in faster formats. They are already in faster formats. Have you seen it? But did you see where I click? I click on spade on database 2, and I click on this thing, view, and it is coming up. Are you on faster formats? It's on Galaxy. You run it on spades, where you have the last thing you did on spade before Proca. Who is there? Who have seen? Have you seen it? No, not on Proca. Go to where you have spades. Spades on Galaxy. Uh, what is that value? Hmm? No, the one you opened before. Okay. And see, you are not go and watch what I'm doing on YouTube. That's what I'm saying. This is the faster format. That's the faster format. Who has gone to the level I got to? You've seen it. That's good. No, why I said so. Okay. Still on stage and I just looking. Follow the. They are still on stage. Are you following me? Follow me. That's where that. And that's where that. You're not be seeing what I am doing on my system. They are not following. And don't assume that they are doing the right thing. And once, broker. How has your own broker come to? Better was what I said. That is, you are not a good follower. You're not a good follower. So, if you want to, what I did was to visualize, hmm, was to visualize the way, and that's why it brought us to. So now, if you remember, for some of you that have started retrieving your sequences before, where you went to NCBI or PDB, you got all those proteins and this one. This was where they also ran it to that level. That's why they put it on database. So now, if your organism is now, so especially for some of us that are running de novo genomic assembly or comparative genomic assembly, so we can also submit this one to NCBI and it will be named after us. So that anybody, anytime, the same way you are also going to that website and retrieve those sequences, people can also go there and retrieve your own sequences like that. So that is exactly what they did. Now, then again, what you actually need from space is the context. But don't mind, let us see the way. So this is the context. This is the context. So this one has actually spades. When you look at the context, spades just pull up all the sequences without any separation. You see, this one has not actually, but the first one I showed you has annotated it. That one has annotated it. That's why I said spades is for genomic assembly as well as annotation. Now, if you check very well, my own proker has finished running. My own, it has finished running. So you can just view, click on view. So let's see, view, one of the proker data sets. View, are you there? View, and let me see what you see. Follow it now on YouTube. Follow it on YouTube. That's where you can be seeing what I am doing. You cannot just assume that when you are hearing me, it's quite different from when you are seeing my own screen. It is when you are seeing my own screen that you will know where to move from where. So when I click on view, I have this data, and I click on the second view. So I have this one. So immediately after, you must have run immediately after you must have run the procar. The next thing, let's go back to our workflow. So that one said space for de novo assembly, and you will use procar for what? For annotation. Procar for annotation. So, and this the workflow has actually given you what is going to be the next, the procedure. So the procedure is that you use spade, that is the Python command. You use spade to assemble. You use procar. You use Bacta to annotate, and you use procar for building what? Building the pangenome. So the rationale is that spade is a standard assembler for bacterial whole genome sequence. Bacta is a modern, actively maintained prokaryotic. Now, let's say you don't know. You can see that on your galaxy, what is coming up when you click your procar is that you do not understand. Just go and screenshot it. Screenshot it and take it. I'm going to take it to where? ChatGPT. So just attach it there. Control V. Can you interpret the attached procar results? Attached, okay, to a layman or beginner. Bioinformatics. So let us see what ChatGPT is going to do. Now, then again, you see one advantage that you have is that, do you know that as you are doing all this, you can be writing your final results, your final reports, your project, you can be writing it along the line. You will just tell, as soon as you are running it, just tell ChatGPT for you to to write the thesis for you. Okay, so what is already explaining to me. Procar is a tool that read your assembled genome and try to identify the genes inside it and guess their functions. Okay, it's now telling us that Procar will try to figure out where the sentences, that is, the sentences here are the genes, what each sentence means, that is, the protein function, and whether the sentence matches anything already known. So it's going to tell you, procar result is going to tell you your gene, where you can find the gene, and it's going to tell you the function of that gene. And it's not only going to tell you that, it's also going to tell you whether there are similar genes like this before. So now I find a gene. I check what other databases say about it. I updated the name so you can get a more accurate description. So look at already. Number one, in a simple language, it is telling you of uncharacterized protein. And that uncharacterized protein is RV2295, which is just an hypothetical protein. So they just predicted that protein. Nobody has ever assigned any function to that protein before. And that's why it's telling you that from the results that we have run on Procar, the interpretation now, if you don't interpret it at all, just copy it and bring it to ChatGPT to interpret it for you. So from there, you will just write out a lot of things about all this. So when you copy it out and you type it, you now tell ChatGPT to expand it for you. So you can now see the essence of you having your own laptop now. Nobody will, he is not going to be, is going to be out of place if you still take your write-up to somebody outside to go and write for you. Yeah? Something that you will pay 20-something thousand for. Now, you have ChatGPT, you have your own laptop, you just do it, you just be compiling it one little by little until you are able to have a full-blown page. If you want to write 500 pages, you can write it. And you don't need to start typing. Just copy it, clean it, data, and there is a tool, Grammarly. Grammarly will help you to refine it in a different way. You can even tell ChatGPT to refine it in a way that nobody has ever written anything along that line. You humanize it. So you can see a lot of things. When I said get your laptop, do this, do that, you can write for your colleagues and you collect money. Abi, instead of them paying 20, okay, pay 15,000. If you run it for 10 people, you know how much it means. I'm only giving you origin, and which has actually, if we can make up for the money you used to buy your laptop. So it has already told you that there is an uncharacterized protein, RV2295. It is still an hypothetical protein. It is not. So there is a gene called this in Mycobacterium tuberculosis genome. Nobody really knows what the gene does. But Procar labeled it as hypothetical, which is just a fancy way of saying we know this gene exists, but we don't know what it does. You can see the uniqueness in running all these things by yourself as it is. So you don't need an interpreter. Again, this one is going to interpret it for you. So you can see the work of. So if you now subscribe for ChatGPT, you subscribe for it, and you pay 10,000, you pay this one, and you are doing all this for others. See, you have not made your money. So then there is another protein that they call putative. It's no longer an hypothetical. This one is a putative. Putative means that it is suspected or likely but not confirmed. So this gene look like it's code for a real protein, but science has not fully studied it yet. And what's the name of that gene? RV2297. Then it also characterized another one, uncharacterized oxidoreductase, which is RV2298, which is a putative oxidoreductase. Abi, this gene probably, but do not definitely perform an oxidoreductase function because it is an enzyme that is involved in oxidation reduction reaction. We think this is an oxidoreductase enzyme, but we are not 100% sure. Then it has also characterized. So it is going to eh, it has actually highlighted the function of Proca results for you. And that's paid as well. You can also screenshot it, copy it, and ask ChatGPT to interpret it for you in the language that you can understand. That it should not be too scientific. It should not be too technical. ChatGPT should know that you are just a beginner in this one. It's going to help you to do that. So it has also given us hypothetical protein, that is, the gene exists, but the function is unknown. Putative, we have a guess about its function. Probable X enzyme, that is, there is a strong evidence, but still not experimentally proven. So, and all this one, and all this one. There's one which is toxin. Toxin, that is harmful molecule. So anyone that is toxin, you cannot use it as vaccine development. You cannot use it for drug development because it is going to be toxic to your immune system. So that means anywhere you see annotated protein with toxin, that means you cannot use it to design a protein, a vaccine. And if anybody asks you why, because it is toxic. In fact, there is a tool that you will by the time we start talking about both in drug design as well as in vaccine development, so that's going to tell you how toxic your substance or your protein could be once it cannot pass the stage of being toxic. For example, I think one of the database is toxic, going to be toxic or it's not going to be toxic. And once it is now um, toxic, that means you cannot proceed with that kind of protein, you just drop it. So those are exactly what and what. Now, again, let us now still go to ChatGPT. Let's now tell ChatGPT, please, are there other unique proteins with unique features or unique functions uh, in my protein or in my Proca results? Proca results. So please tell me. Okay, can you see? ChatGPT is already telling us. You see? Are you there already? You are not there. Who is following you on YouTube? That is the result. You can see. So you can see the uniqueness. You can see the flow of or the sequence of what we are doing. You don't need. You see, I was listening to this thing. He said, you see, the world now, if anybody wants to deceive you, that's right. If anybody is willing to deceive you, you know what the person will tell you. Person will tell you, see, this laptop, get laptop, do unnecessary. This guy is just stressing you. Now they said AI, artificial intelligence, from 2026. Do you know what they say? from 2026, if you want to know anything at all, you should learn AI. Learn it. And what we are using, all this what we are using, AI now. It's machine learning. It's AI. So anybody that does not know how to do this, they said this year, those that are of age 20 to 25, AI has taken more than 15% of the job it's supposed to do. For example, in those days, that is when you need personal assistance. Nowadays, we have AI that can run your diagnosis. Hello, please, I am feeling dizzy. I have this cough. I have this, that. I have this, that. Can you guess what could be? So you think that, probably you need to rest. Probably you need an exercise. Probably you need to combine this kind of diet. And this is exactly what somebody somewhere can hire you, as somebody who's in Nigeria can hire you, and you now be seeking for your own advice. The person will be thinking maybe you are a magician. You are not a magician. What you know is artificial intelligence, and you know how to make use of it to do a lot of tasks. You know one thing, one day, do you know that one day I did this thing, architectural drawing of one building, very big building for somebody, and I used ChatGPT to do it. I'm a biologist, and I did the work of somebody who is there. The person just said, I just want you, can you pay my money? I will pay now. That day, I think he paid 25,000 or something. It was a very small job. Within 1 hour, I got my pay. So what I'm saying in essence is just that people that do not, and that's why I do not query people. If I don't know something, I will say I don't know it. It's not that it is not possible. Gone were those days that you need to start racking your brain. How do I? You'd see in those days now that students, they will, if I had project students, they will bring their topic more than 10, 50, 70 times. Me, I don't. Now, do I even have time to even go through, check their punctuation mark, tell this, tell that? If you bring it, I'll just put it there. Please help me to punctuate everything. It's going to do it for us. In fact, there is a that Grammarly. I'm going to subscribe on it to this level that we call it a writing assistance. I'm going to check my grammar. It's going to check the punctuation mark. If I say, as a PhD student, help me review all this and bring out. I'm going to refine it and bring it for you. You think I'm a magician. You think I am. So what we are saying in essence is, nowadays, the only thing that you can suffer from is if you don't have good laptop and internet. Good laptop and internet. That's why me, I will always be an advocate. As soon as you are coming to be, in fact, to me, it's even a shame for somebody who's in HND without a laptop. That is it now. Nowadays, we have people students writing WAEC. With what? Laptop. With system. Data science, data this thing. They can do a lot of things for you. So, and you now be a big sister. You now say, yes, now, at least I'm not your mate. I'm your 10 years of kidney. I'm older than you. And you are older than me. But if you are older than me, what do you have that I don't have? Do you know how to work on laptop? Do you know how to use artificial intelligence? Do you know how to do this? And you know, I don't know. I don't know. So what we are saying in essence is that now you now have what this one is telling us is that we have PPE family proteins, which are uniquely mycobacterial. That is, it is only them that you have there. And PE23 is an example. What this means that these proteins are unique to Mycobacterium tuberculosis. So now, if you have two species of Mycobacterium, we have Mycobacterium tuberculosis and we have Mycobacterium bovis. That bovis is found in cow or cattle. It is found there. Now, if you now see, when you now run Procar and you now see PPE or PPE family, that is to tell you that it's only in Mycobacterium tuberculosis that you can have it. If you are not, if you cannot get it there, that means you are not dealing with what? Mycobacterium tuberculosis. You are dealing with a different protein, I mean a different species entirely. So another one is a coenzymes. Now, if you now still ask ChatGPT, please just tell me from the Procar results, please extract, should I tell you? But I should not tell you. Say it will not teach you how to be lazy. Just tell ChatGPT, please extract 35 proteins with their faster formats that I can use for my vaccine design or drug design. It's going to bring it for you. You don't need to start going to NCBI by the time you do this, by the time it's going to bring it for you live and you continue your journey. So that's why I said, if you have your time, you can finish the rest of your project in the next 1 month. Or everything you have been labored with, all this while, you will finish it. Finish it within the shortest period of time. So, where are we? We have run, let me see, Galaxy. Yes, that is Procar results. So after Procar, I think okay, please run, let's run Procar up to that level. Continue, run that Procar, run your own, run Procar on your own results. Hello? Ṣé bẹ́ẹ̀ yẹn nìkan ni kíní wà ní? Bẹ́ẹ̀ náà ló fẹ́ kú. Bọ̀tá ò ní yọ̀ mí. Tá ò ní yọ̀ mí. Tá a lọ pé ẹ má ti, wọ́n ní a máa ń sáré. Hello? Why is this? What happened? That happened.